Io tri, io guarda, guardare un viso che fare. Non sa pas dire que non sa pas carregar de tudo. Vai para aqui. Cerco di cerco di cerco di yes.
वो मगर फिर चेंज कर लेना, बाद में चेंज कर लेना, या दे दी मैंने।
उनका बताऊँ तो मीठा हो जा उन तक उनका
हाँ।
Morning participants, can you hear me?
Yes, sir, you are audible, sir.
Good morning to all of you. We have two sessions today on the tutorial section. One is based on the virtual tour on the food analysis, another is the laboratory demonstration. We are having today mostly to demonstrate the type of instrumentation which are being used in the probiotic research. That will be very important, whatever we are having, and that is available. So, in the tutorial session, we are dividing into two sections. One is based on the ability of understanding the earlier lecture by Professor Chikitas. We had two lectures yesterday, and if you have any doubt or clarity, we can discuss this even in the tutorial section today. So the tutorial section will be based on mostly the lectures delivered by foreign expert, and also what are the instruments which are being used to understand these probiotics to perform the experiment in the laboratory. We try to show you that instrument using the demonstration technique directly connected from the laboratory. In some of the tutorials, we are also taking you as a virtual tour to the industry. We have contacted to Yakult India Private Limited, which is an industry which is based on completely on probiotic application. You might be knowing about Yakult, and where they have used certain probiotic bacteria, and they are selling as a product to different industrial level and the market. So it's a kind of learning you can have with this. So with application point of view, if you see the application, there are certain things which are applied. This probiotics can be applied mostly in the food system. So we have connected to an industry called an Encrom. They are very much efficient in analyzing the food system using different instruments. So we will have this idea from there that how food can be analyzed to understand their application using different probiotic system. In another half of our tutorial, we'll take you to the laboratory of microbiology department here in Barcelona University. There they will demonstrate you what kind of instruments can be used for growth, for purification, for detection, for identification of these. I think you can unmute the participants if they have any clarity on based on the previous lecture, they can ask. So all the participants, let anyone want to interact on their suggestion or their views or anything, if you want to add on the lecture delivered by Professor Chikindas, or anything technicality, if you think that it was not audible or something, any issue, you can discuss. So the lecture one was mostly based on the criteria of selection of probiotic bacteria. That is very important. What he delivered yesterday is how these probiotic strains can be selected, and what are the sources you can identify from where these in the environment, you have a different sources. You have a food, you have water, you have a soil. All these sources are actually visible, and one can hit what kind of things he can go on for the collection of these samples and identification and isolation of these strains. So that is very important, number one, what you pointed out. And after selection, it is very important to identify it. See, if strains are unidentified, then you don't know whether it is Lactobacillus enterococcus or E. coli or anything. So there are techniques which identify to the best of its capability that what are the features you can use for identification. So number one, that how strains can be identified. You can use morphological behavior, you can use biochemical behavior, you can use molecular technique, for example 16S RDNA sequence for identification of the strain to the best. And once the strain is identified, then it has to be characterized for its application in the probiotics. This is what he delivered. That identification is one of the most important, and then it is a features. What are the features? He put the safety aspect on the top. That the strain what we are using is safe. Right. So what I thought that in the previous first lecture, it was mostly on the property of the strain, that is the criteria for the selection. What criteria has to be taken care of? For example, these criteria like ability to survive in the acidic environment, ability to survive in the gut environment, ability to adhere the cells, that is autoaggregation or co-aggregation with the other bacteria, so that by autoaggregating it adsorbs on the surface of the epithelial cells to perform the function. Whereas this antimicrobial activity, by showing antimicrobial activity, kill the pathogens. So these were the some highlights of the previous lecture delivered by Professor GIAN. If anyone want to interact with this lecture or any doubt, we can discuss. So no one want to ask anything? Any clarity, anything you want to ask, or any question which was not covered yesterday? this is a tutorial section, it has to be interactive. We have to learn, you have to understand this by interaction only. If you are not interacting, then we cannot perform this tutorial section. We are waiting for a person to connect with the laboratory team. Meanwhile, we are solving these tutorial issues with the previous lecture. Am I audible? That's fine.
Yes, sir.
Okay. So it is only 38 participants till now.
39.
Right.
39, 38, sir. 38 participants.
So there are some instructions for the participant. I think all of you have seen the brochure. If you see the brochure, the schedules are clearly given that there are 10 lectures and 10 tutorials divided into different dates, starting from 21st and the last lecture we have on 27th, right? And on 25th we are going to have a tutorial in the morning. On 25th, it is Saturday, and 1 hour exam in the evening. And as per the GIAN guideline, the participant has to pass the exam to get the certificate. So it is my humble request to all of you, kindly join each lecture and tutorial. The examination will be based on the lecture delivered by our resource person and the tutorials which are going to be conducted during the session. So you will learn those techniques and the theory, and those things will be asked during the examination. You have to pass it, you have to attend the examination. That we cannot ensure. If you are not able to attend the examination, we are sorry, we will not be able to provide you certificate. So, in this section, we are going to have interaction with you based on the practical aspect, what can be done by selection criteria, what can be done by identification, what can be done on the biosafety level of these probiotics. Like in general, the criteria for selection of microbes, as Dr. Chikinda's study was suggesting that each microbe, they have different behavior. So from one strain to another strain, or one strain itself, if it goes to the one human to another human, from individual to individual, its behavior changes. So it is very important and has to understand that what are the ecological niche has to be selected, which are having probiotic strains, and if you select those, then you can go for the identification, isolation of the strain. So as far as his concern, the theme was that the selection or identification of ecological environment from where you can do it. So it is found that most of the fermentable product, either it is in the nature or it is in the food, most of the fermentable products, they are having the growth of certain lactic acid. And out of probiotic microorganisms which are being used, almost 80%, more than 80%, they are lactic acid. Some of them are bifidobacteria, some of the bacillus species are also used. Others are yeast, Saccharomyces species. Yeast is also used for the probiotic application. But if you think of bacteria, mostly they belong to group of lactic acid bacteria. Most of the probiotic bacteria we are having in the commercial application, they are under the group of lactic acid bacteria. Now you have to think of that which ecological system allows the growth of these lactic acids. Right? So if you think that way, that what are these area where you can go for selection? So number one, you have to target those which are fermentable. Fermentable means any plant material which are like this. That is called as silage. S-I-L-A-G. Silage is something which plant-based material which supports the growth of bacteria in the moisture conditions, and that allows the fermentation. So one can go for isolation with the silage, one can go with the rhizospheric root in the plant system and isolate it. That I'm talking about the non-conventional, which are non-food based. In food, you have a different variety, different options for isolation. But what I understand that when a student go for isolation of probiotic bacteria, the most and utmost features he should utilize to get the novel strain, the strain which does not exist somewhere in the database. Then you will be the winner. That you are having a strain which are something which is very, very important, and the feature which shows that this strain is not available in somewhere in the database. Right? So based on that criteria, you can use it. So if a student goes in India and students in the US or Australia or anywhere else, then that depends on you. With the soil in India, we have differently, soil in other globe of the earth, we have differently, and they allow the growth of different microorganisms. So by chance, by luck, in India, we are having different variety of cultural environment. So if you see the western in India, their monsoon is different than the eastern, and the north monsoon is entirely different than the south. So this you have a very much diversity in the flora and fauna because of the climatic conditions are different, and that climatic condition actually allows the growth of certain bacteria there. And if one hit these climatic regions, definitely these will be different than others. So that is number one. If you want to isolate from the environment. Now, if you think of that, if you are not able to isolate from the environment, what we can do, that most of the food which are mostly cooked in the fermented foods. So these fermented foods are generally having lactic acid. You can name these fermented foods. Like anyone can say fermented food. What are the fermented foods you use in daily life?
fermented foods are curd है, beer है, जो रबड़ी बनाते हैं वो भी fermented food है.
Yes. Anybody
गर्मियों में
else?
बनाते हैं.
हाँ जी, जैसे इन्होंने बताया रबड़ी, it is very good, because of the. I told you that you have to isolate a novelistic. So you have to think of foods which are not explored till date, because those foods which have been explored, their probiotics are already characterized. We can take those foods which are not studied. So anyone can tell the food which are fermented foods used in daily life. बताओ भाई, ये आप 40 के 40 लोग बैठे हैं. See, we have to make this program attractive and interactive. Not by just listening, but also by putting our idea to make it more interactive, so that it helps the person understand only theory, not only, but also work. Yes, dahi, yogurt.
The cheese is there, yogurt is there, curd is there.
Yes.
Vinegars are also there, I think.
Yes.
White vinegars are also fermented. Alcohols,
Yes.
most kind of the alcohol, they are also fermented.
Yes. Although idli, dosa, sambar, vada, and most of the South Indian foods all are based on fermented. Why I am going to list all the, you might be asking, what sir is asking. We know it already, it's a wastage of time. No, I am digging this information so that any food item which comes out of your knowledge, which is not explored till date. Explore means which you know. Explore means all those on the curd based, on the cheese based data, on the dosa based, on the South Indian food. All these people have worked out mostly. What one said, like Swaraj is saying that rabadi. I think if you go South India and say someone's rabadi, he will not able to understand what you are talking about. It means it is an indigenous food of Haryana, and very less study basically on the composition of probiotics. It is available very less. I have found some of the papers they have worked on rabadi, but not much data is available. For example, if you isolate a strain from the rabadi and propose to the commercial level that yes, what is the importance of rabadi? And in Haryana, people understand. Dr. Mehak might be knowing, Swaraj also might be knowing that how rabadi is prepared. You add some chach, dahi in the flour. You use some flour. So there is a combination of plant product, flour with the curd, with the chach. What you are mixing? You are mixing one is the probiotic ingredient, that is your dahi or chach or buttermilk, whatever you use. That is the probiotic content. But why we are using plant-based product like flour, or you can use flour of different grains like bajra or atta, wheat flour. So these flours contains prebiotics. The prebiotics from the plants, and those prebiotics are actually helpful for the growth of probiotics. Prebiotic oligosaccharides which are found in the plants like wheat bran, rice bran, they are very rich source of even millet brans. All those brans are very rich source of prebiotics. And trust me that if you read it, you will find that prebiotics are the compounds which supports the growth of probiotics and not digested by human gut. So for human gut, it is not required. Having prebiotic and probiotic both. So these things has to be explored. This is since dawn of our civilization. Since the past, people are using such kind of food. Uh, it's just by chance they have mixed both plant product and dairy product. Just to having prebiotic and probiotics, not earlier based on the chance, but now it is a clear scientific evidences which suggest that we are using combination of prebiotic and probiotic together for the growth of this probiotics in this food. And if you consume this food, and gut will be healthy, and its modulation to the different organs, different system will be much better as compared to those who are not taking these probiotic foods. So this is what reasons I'm asking. Maybe students, some students are outside from Haryana also, they are joining. If they are knowing any food item in related to rabadi, something one food is in Bihar. They are using some sattu. They mix sattu with the dahi or charge. You can say it's something like a rabadi, but the process in the hot season. So, I mean, those kind of foods, if you are going to select a strain, suppose you are going to work on the strain, you have to select those ecological niche, ecological environment where these probiotics are present, but not tapped by the community till date. And you will be the first discoverer for this strain if you go for. Like if you go